Welcome learners. The topic for today is mechanism of insulin signaling. To get into the basics, a significant stress faced by all animals is the unpredictable cycle of feeding and then starvation that ensues between meals. Humans solve this problem by storing nutrients in forms that can be used as energy sources during periods of fasting. The driving force that regulates this process is insulin, which is a hormone released by the beta pancreatic islet cells in response to elevated levels of nutrients such as glucose in the blood supply. Insulin binds to its receptor on the major insulin responsive tissues of the body, namely skeletal muscle, adipose tissue, and liver. This triggers the activation of a signaling pathway whose function is first to stimulate the transport of nutrients such as glucose, amino acids, and fatty acids from the blood supply to these tissues, and secondly, to promote the conversion of these nutrients into storage macromolecules such as glycogen, protein, and lipids. Failure to regulate the uptake and storage of nutrients efficiently following feeding results in diabetes, which is estimated to affect approximately 2% of the world population. So this is the diagrammatic representation of the receptor tyrosine kinase. This insulin receptor is a heterotetrameric complex membrane spanning receptor, what we call it as receptor tyrosine kinase RTK, made up of two alpha subunits and two beta subunits that are disulfide linked. The insulin molecule binds to the extracellular alpha subunits which activate the intracellular tyrosine kinase domain of the beta subunit. Insulin binding to the receptor tyrosine kinase results in a series of actions like dimerization, autophosphorylation, activation, and finally signaling. In dimerization, the binding of insulin to the alpha subunit of the insulin receptor causing binding of two receptor tyrosine kinase receptors, which is called dimerization. This process is crucial for activating the receptors as the binding induces conformational changes that bring the intracellular kinase domains beta subunits into proximity. This proximity allows for trans-autophosphorylation. Autophosphorylation. The proximity of the dimerized receptors allows for trans-autophosphorylation, where each receptor phosphorylates the other on specific tyrosine residues, and this phosphorylation is essential for relieving cis autoinhibition and transitioning the tyrosine kinase domain to an active state. Finally, the activation and signaling. The autophosphorylation of specific tyrosine residues creates binding sites for signaling proteins, leading to assembly of intracellular signaling complexes. These complexes propagate the signal through various downstream pathways such as MAPK, PI3 and PKC pathways. So this is the diagrammatic representation of tyrosine kinase receptor and the processes that is happening after binding of the insulin to the receptor like dimerization, autophosphorylation and activation of the downstream signaling proteins. To dwell into the details, the insulin receptor binds to phosphorylated residues on proteins, namely a family of large docking proteins known as the insulin receptor substrate family IRS1, as well as the adapter protein SHC SRC homology 2 domain containing. When insulin binds to the extracellular alpha subunits of the insulin receptor, a conformational change is induced. which then results in the autophosphorylation of several tyrosine residues present in the beta subunits. These form the binding sites for IRS proteins, which contain phosphotyrosine binding domains (PTB), or for HSC adapter proteins containing SRC homology 2 (H2) domains. Binding of the insulin receptor to either IRS or SHC forms a platform that allows for the assembly of a signal transduction particle that gives rise to multiple intracellular signaling pathways. Two principal pathways result from the insulin receptor IRS interaction: the PI3 or AKT, also known as protein kinase B (PKB) pathway, and the RAS-MAPK, also known as extracellular signal regulated kinase or ERK pathway. We are going to discuss on PI3K, that is phosphoinositol 3 kinase pathway, as it is linked exclusively through IRS and is responsible for most of insulin's metabolic effects in the cell. The PI3K pathway is activated by the binding of PI3K regulatory subunits P85 to IRS1. This results in the activation of PI3K catalytic subunit P110. Once the P110 subunit is activated, PI3K then catalyzes the phosphorylation of phosphatidylinositol diphosphate to generate PIP3, that is phosphatidylinositol triphosphate. PIP3 is an important second messenger that functions to recruit PDK1, that is phosphoinositide dependent protein kinase 1, and AKT to the membrane, where phosphorylation of PDK1 activates the serine threonine residues of AKT. From here, AKT plays a critical role in downstream process. AKT acts in the regulation of glycogen synthesis via glycogen synthase kinase 3 (GSK3), another serine threonine kinase. GSK3 is inhibited when phosphorylated by AKT-PKB, which results in glycogen synthesis. AKT also regulates the translocation of insulin-sensitive glucose transporter GLUT4, which is sequestered in intracellular vesicles of muscle cells and adipocytes to the cell membrane via exocytosis, where it facilitates the uptake of glucose from the blood into cells. This is achieved through the phosphorylation of AS160, a GTPase activating protein that activates RAB, a small G protein involved in membrane trafficking, by blocking the exchange of GTP for GDP. The final is the pictorial representation of insulin signaling cascade. To summarize, the phosphorylated receptor recruits and phosphorylates the insulin receptor substrate 1 on tyrosine residues, which then recruits dimeric PI3 kinase via SH2 domains on P85 subunit, which in turn recruits P110 subunit that successfully brings the substrate PIP2 to the vicinity of PI3 kinase. PI3 kinase catalyzes the phosphorylation of phosphatidylinositol biphosphate at the plasma membrane to PIP3, which then recruits PIP3 dependent kinase, that is PDK1 and AKT, allowing PDK to phosphorylate and activate AKT. Activated AKT phosphorylates and inactivates the AS160 and RAP10 GAP, which plays a critical role in trafficking of GLUT4 storage vesicles to the plasma membrane and surface expression of GLUT4. Have a happy learning. Thank you all.